very quickly over the exceptions to Mendel's law and more exceptions to Mendel's law, and I was supposed to come back to it, but what I actually thought is it's better for me to do this after I cover this whole the chromosomal basis of heredity and mitosis and meiosis, and then I thought that'll make a lot more sense because you sort of get why some of these things are happening. And so today I thought I'll start with cells and then how they divide. That's really going to be the main topic for today. And this will actually help you understand why Mendel's laws and their exceptions work the way they do. Okay? So I don't know if how many of you have gone over cell, the basic idea of cells. I think 7th and 8th graders may have already some introduction to it, right? But yeah, what I'll do is I'll just go very briefly over the most important parts of cell. So cells are the basic building blocks of any living organism, and I'll only go over stuff which is important for heredity, and they exist in all different forms. Us human beings, each of you are a collection of a bunch of cells, okay? And these are some of the examples of different kinds of cells. The organs that we have are really a collection of different cells, and we are really a collection of organs which are all connected together by a network of nerves or blood vessels or whatever, different things, and they are all cells. So essentially we are a collection of cells which are also connected by other cells. And all our tissues and organs are made out of cells. And there are two kinds of cells, prokaryotic and eukaryotic. All of us, human beings, animals, most plants, they are all eukaryotic. Prokaryotic are things which are more like bacteria and viruses, mainly unicellular organisms. So we focus more on the eukaryotic cells because all the heredity we have studied till now, actually it's in the context of eukaryotes. And I'll skip this. This is how a prokaryotic cell looks like. This is really how a eukaryotic cell looks like. And the important things that you should note here is that every cell has a cell wall or a cell membrane, and inside the cell there is something called a nucleus, and the nucleus contains all the chromatoids, which are really the basic units, the basic physically they are the things which are responsible for the traits and characters and genes. So the genes are actually physically part. Sorry?
So they're called chromatoids?
Chromatids.
Chromatids.
So or chromosomes. Yeah. And as you see here, this is the nucleus, and the chromosomes are all lying inside the nucleus. And the other important thing to note is that there is something called the centrioles, and they play a very important part in cell division. Actually, they are called the centrosomes, and centrosomes actually contain centrioles. This is a little bit simplified picture. The rest, for now, you can just sort of ignore. And then this shows the different things. And so right there, you see this whole coiled substance inside the nucleus. Those are the chromosomes. Right now it's uncoiled. This is like a bunch of hair that's just floating inside the nucleus. And I'll skip all this. If you want, you can look at it. So the cell membrane, it's really the entrance and the exit. This is where all the nutrients come in and they get absorbed inside the cell. They are then used for making the cell grow, the metabolism of the cell, and then essentially this is your food for the cell, the path for the food for the cell. And it also protects you from other foreign objects coming in. And in the nucleus, there is this chromosome which is made up of DNA, and they are the ones which actually have physically they have the genes in them. Okay. And there is some, and the nuclear membrane is just within the cell. The nucleus is enclosed in another membrane, so that the stuff that lies within the nucleus doesn't just disintegrate anywhere in the cell. It's all contained by this nuclear membrane. And what is the cell membrane's function? You know this, number A. And then the cytoplasm, this is the stuff that lies outside the nucleus but within the cell membrane, and it's like a jelly-like substance that kind of fills up the cell, and it has all the other bodies like mitochondria and Golgi bodies and whatever else is there in the cell. And then I'm just going to skip all this. It's all interesting stuff. This is not about cells, but it's a pretty nice set of slides, and you may just want to review this even if it is for other reasons. But these are the key things about the cell. So just to recap, we have, and we'll focus on only these things now. Okay? We have the, where are the?
It's right there.
Okay. So we are going to focus on a few things. We are going to focus on, and there's the nucleus, and then inside these, I'll draw it with a different color. I don't have that many colors, but actually I should draw this with a different color. So this is the nucleus. Inside this is the nuclear membrane. Then right here, these are the chromosomes. They are right now, these are like strands of hairs, and they just lie kind of in this state normally. Okay? So there are two ways by which the cell divides. And why does a cell divide? Yeah?
To make new cells. And
Yeah,
to multiply.
sure. So, yeah, that's essentially right. So, I mean, people grow, right? You are growing. So something has to happen for you to grow. So part of that is accomplished by cell division. Cells also die. So when cells die, they have to be regenerated. So the mechanism by which cells divide is called mitosis. And when cells divide, they make identical copies of themselves. Okay? And so mitosis, I think, is a Greek word. It means threads. I forgot. But essentially, the challenge is from this cell, you really want to come up with identical copies of this cell, right? And so this happens in, maybe I should, I'll put on the slides later, but I'll go through it on the board first. So you'll see a picture like this in the slides and the notes. You'll see a picture like this. So this is called mitosis, and then this is called G2, and then S, and then I think actually this is G1. So G1 is growth 1, S phase is an intermediate phase, and this whole thing is called the interphase. So just for getting a sense, and this is time, by the way, this is the sort of the life cycle of the cell. When the cell is born, it goes through a growth phase, and then it goes through a S phase, and then it goes through a further growth phase, and then it divides, right? And then it becomes two cells. And this phase is actually the maximum time spent by a cell.
What does that say? Does that say mito?
Mitosis. This is mitosis. Yes.
So what's that? What happens in the S phase?
Yeah, I'm going to go over these phases. So G1 phase, the cell just grows, okay? It just gets nutrients, and then it adds more substance, and it physically grows in size. So now, before we go to the S phase, we should focus a little bit on the chromosomes, right? So in most of the organisms that we study, Mendel's pea plants, human beings, mammals, unless we are talking of some organisms which are sort of not the normal ones, they always have two copies of the chromosome. Okay, and you might remember from the picture, what we did earlier related to monohybrid crosses. You had a T and a T, right? So this T is actually embedded in one of the chromosomes. It's part of the chromosome. It has proteins called DNA. Chromosomes are made out of DNA, and part of that actually tells whether it is going to do a T, tallness of the plant. And that is physically called a gene. Okay? So if you look at a chromosome which is like this, so there'll be a gene 1, there'll be a gene 2, there'll be a gene 3. So these are actually some stretches of DNA, and this particular gene 2 may actually represent the tallness of the pea plant. And so a chromosome has all these genes, and then there are two copies of this, right? Remember? So what does that mean? It means that there is another chromosome here which also has gene 1,
Gene 2, gene 3.
gene 2, gene 3, pretty much at the same locations, but the content of this gene may not be the same as the content of this gene. Just like we had this T and small t, this could be one allele, this could be another allele of the same gene. And physically they are present in two different chromosomes. And for most of these organisms, we are all diploid. Diploid means we have 2 times n of these chromosomes. So let's say this is chromosome 1, and they come in pairs, right? So these 2 chromosomes is called one chromosome pair. There's another chromosome that's let's say has gene 5, gene 6, and so on. And actually I'm just making this up. These genes are something else. But the idea is that these come in pairs and we are called diploid. Okay? So now what you have to do in mitosis is really to actually duplicate this whole thing. And think of the nucleus. It's got essentially, if I were to draw this in different colors, so this would be, let's say, one chromosome, this would be its sister chromosome. Like basically, this is one pair, and then you probably would have another pair like this, right? They're all kind of wound up like coils of hair, okay? Now, what you have to do is you have to make exact copies of this. And this is almost like very, very fascinating the way this happens. And you get to realize that your cells are so sophisticated. They are just amazing, the way they do this. So what happens is, in the S phase, the precursor to the division happens. And you come up, the cell actually produces another copy of this, and this is attached in the middle like this, okay? And this is still in this uncoiled state, but each of this grows another copy. So now, let's say we are human beings, right? We have, this n happens to be 23 for us. So we have 46 now. 46 chromosomes, 23 chromosome pairs. We essentially now end up having 46, this is still called 46 chromosomes, but each chromosome now has essentially 2 identical copies, and they are called sister chromatids, okay? So, and they look like this. So this thin strand of hair, which was the original chromosome, there is another copy of this, and they are joined here, and this place is called the centromere, okay? Yes?
I was confused on the meaning of diploid. Can you explain that again?
Diploid means that every chromosome has a pair, has its corresponding pair.
Yes.
So that's where, so basically you have this is where remember, every cell has one copy of a tall and maybe one recessive version and another like dominant version. Those are physically these things.
Oh.
So don't get confused. Before the S phase, there are no sister chromatids. There are actually 46 chromosomes, which is 23 pairs, and each pair has the same genes, but potentially different alleles. Is this clear to everyone? Okay, this is very fundamental. Okay? And that's why it's called diploid. The DI comes from the fact that it's 2. There are organisms which are triploid. There are like 3 copies, and I don't know. I mean, in biology, everything is possible. Okay, that's the rule. So now you have the sister chromatids. So at least something has happened. Some division has happened. But it's all still like this. And then it goes through a further phase, okay, of growth. And then the real mitosis happens. And we'll see what happens in the real mitosis. Now, this real mitosis is actually comprised of 4 phases. And then there is a 5th phase, which is actually probably not part of mitosis. It's called cytokinesis. And these are all there in the slides, so you can still take the notes, but there are a lot of terms in this mitosis thing that we'll go over. Cytokinesis. So in the old days, okay, there were no diapers. So babies were just left on these little like cloth mats. And what do you think they did after some time? Probably pee, right? So those used to be called pee mat. Okay? That's why this should stick to you. Pee mats. P stands for prophase, M stands for metaphase, A stands for anaphase, and T stands for telophase. Okay? It's very hard to remember this, but if you remember this P mat and the baby peeing on a mat, you will remember this, okay? Yes.
Wait, can you repeat what stands for what?
Yeah.
Okay, P stands for prophase. M stands for metaphase. A stands for anaphase. And T stands for telophase. Remember this, the phase is always there, and then I think the rest will come to you. You just go over the slides a couple of times. Okay? And this whole thing happens for probably 20 hours, and then this finishes in 1 hour. So this is a very small percentage of time that the cell spends actually doing the duplication. Most of the time is spent doing the growth. Okay, but some of the stuff has already happened. From the, say in human beings, you had 46 chromosomes, you already got effectively 92 chromosomes, even though we still call it 46 chromosomes, but now there is an identical copy of each chromosome which is like attached in the middle through the centromere. And by the way, depending on where, it doesn't need to be attached in the middle. It could be attached towards the end or towards the very end, and depending on where it's attached, they have come up with different names. This is called metacentric, this is called acrocentric, and this is called telocentric. Okay?
You have to remember those.
You have to read this because but just to get this, there are names for where these are attached. And the main idea is that they don't need to be attached here. They could be attached Another one could be attached. This would be called metacentric. This one would be called, I chose a different color, I shouldn't have. This one would be called acrocentric. And this one would be called telocentric. And these things are called centromeres. Wow, a lot of terms. I can't do anything about this. This is how they have done it in the books. Okay? So now enter mitosis, and the first phase of mitosis is called prophase.
Metaphase, anaphase, and prophase.
Okay, what happens in prophase? A few things happen. This is where the strands of chromosomes actually start coiling up, and they look so. And just to give you an idea how big these are, if you were to magnify this by a factor of 1000 to 1500, this is about the size it will be. So it's very clearly visible in microscopes, even though I haven't seen one. I only read in the books. So, but I'm guessing you will probably see one pretty soon, or probably already seen one. So, and if you look under the, yeah.
What happens at prophase one more time?
Prophase, yeah, I'm coming to that.
Oh, you see prophase.
Yeah. So I'm just saying that you wouldn't be able to really distinguish the chromosomes if this was before prophase. You wouldn't have been able to know that these sister chromatids have formed in S phase. But once it enters prophase, a couple of things happen. This is the cell, this is the nucleus, and now you will be able to see the chromosomes very distinctly. So this is now one pair of chromosome, even though, and this is the centromeres. So what has happened is, these chromosomes have, which were all uncoiled, have suddenly thickened and coiled together, almost like a spring, and now they are all visible in this form. Okay, this is what happens in prophase. And now, don't get confused. These are the centrosomes. These are the centromeres. I'm myself confused. There is something called centrosomes, which you saw in the cell. Actually, they were called centrioles in that figure, but actually these are centrosomes. And the centrosomes, they are also in the nucleus. They also separate. So you have a centrosome, which now two copies of the centrosome form. I should put that in a different color. So let's take an organism which is got 4 chromosomes. Okay, 4 chromosomes. I'll use some other color. So in prophase, these 3 things happen. One is the centrosomes separate, and the thickening happens, and this nuclear membrane starts disintegrating. Okay? But the chromosomes are still kind of spread around in the nucleus. Enter metaphase. In metaphase, something magical happens. Okay? These centrosomes go to 2 different poles. Okay? They travel and then they set here. So let me draw another picture. So this is the prophase, and this is sort of breaking up. And I'm going to right now leave the rest of the cell alone and just draw the nucleus part. So now the membrane is gone. Okay, what you have is the centrosomes sitting at opposite ends, and then this is the magic. The chromosomes go to the center of the cell, the equator of the cell. What was the color?
Which phase is this right now?
This is now in metaphase. Okay? And then some spindle fibers start growing. These are like strands of hair. And inside the centrosome, you have the centrioles. And the centrioles are like, the things that we use to pull thread? Like
spindles.
spindles, yeah. They are like spindle. And these are like little threads that are growing on the spindle. Okay? Amazing, right? And then these go and attach exactly to these points. And not just that, there are others that sort of get connected like this. And so this is what happens. Okay? A lot of threads grow, but there is always a thread that goes and attaches to the centromere of the chromosomes. Okay? Okay, now we come to anaphase. What happens is the spindles start pulling. Okay? And so what happens is because these also push against each other, these threads. So this creates a force to separate this out. So it's like almost like having this thing and putting two strings here and then applying force and the two separate out. Right? Amazing. And then these in the next phase have moved towards they have separated, and now you understand why there were these sister chromatoids, right? Because for two cells you need two copies, right? And then this comes, and then there is no nuclear membrane, by the way, at this point. It's all in the cytoplasm. And the threads are still connected and they are pulling. And then finally in the telophase, actually two nuclear membranes start forming, and this now really starts looking like, and this is one of the reasons I didn't do biology. I couldn't draw very well. But and a nucleus has formed here.
Could you please move up the screen?
And the nucleus has formed in the telophase, and essentially now you're back to exactly how you started off with in the nucleus. So you have, in the coiled form, you have the chromosomes, there is a nuclear membrane formed. This is sort of the last phase of mitosis, and this is followed by cytokinesis, where the rest of the cytoplasm and all the other apparatus that's there in the cell, and there are a lot of things in the cell, they all separate out. They make copies of each other, and it's called cytokinesis. And then essentially you have a copy of the cell. Okay? That's what mitosis is about. There are very good videos in YouTube. You can also go to Khan Academy and watch this. But, yeah.
So is the splitting the telophase? Or which one is the telophase? Because I'm confused.
The telophase is where this
Okay.
shape
Everything
forms,
is
the nuclear membranes form. The actual splitting happens in anaphase. In metaphase, it lines up in the equator and the threads have attached. And then in anaphase, they actually. And then in books, you'll find sometimes they go to even finer level of detail, but it's not really important for your level to go at that level of detail. But what's going to happen is, in Science Olympiad, they will give you a picture and ask you what phase this is. Okay? And you should be able to say, now that you understand the concept, we'll go over the pictures in the slides, because I didn't do a very good job drawing them. But at least hopefully you got the idea, right? Okay, good. Okay, so we have mitosis. So now the more interesting stuff. It's called meiosis. So meiosis in Greek means reduction. Now tell me, if you have a diploid father and a diploid mother, and you just had mitosis, the kind of thing that happens in mitosis, if the two came together, what would be the problem? If you just took two cells from the parents and put them together, what would be the problem? Devangi?
They have different cells and different genetic material.
Yeah, but
Oh.
there is something more fundamental that will go wrong. When two parents, you saw that in Mendel's laws, right? When the two parents come together, actually you get genetic material from both places in equal proportion. But my question is a little bit different. Suppose you just took a cell from your father, and a diploid cell from your father and a diploid cell from your mother, and just formed a new cell. What would be the problem with that? It'll still have the genetic material from both parents. Yes, Srija.
Both are diploid, so if one fertilizes the other, then the resulting would be four, like quad
Exactly.
something.
Exactly. That's exactly right.
Can
So,
you explain that?
did you get this?
No.
You have 2n chromosomes, right? Here you have 46 chromosomes. You have 46 here. If these come together and fuse, you'll have 92 chromosomes, right?
Uh-huh.
And in the next generation, you'll double it up. And soon, it doesn't work, right?
Oh.
So that's why you need something else. You need a reduction, right?
23.
Exactly. And so these are special cells. These are not the cells which happen in your skin or your brains or your legs or your muscles, other muscles. These are called the gametes, and these are special cells, and the process by which they are formed is called meiosis. Meiosis still starts with a normal diploid cell. Now, it's a special kind of cell because not all cells undergo meiosis. Only cells which will form gametes undergo meiosis. What's a gamete? A gamete is a cell which is special in the sense that it's got half the number of chromosomes. And so, if a gamete from a father and a gamete from a mother come together, it will again form a diploid. And all of us, all human beings, all animals are a result of, this is called the sexual reproduction, where two gametes, one from each parent, comes together and forms a new organism. Okay? And the process through which the diploid cell gets reduced into this haploid state, it's called haploid, is called meiosis. Okay? Here? So now the problem is a little bit different. You have 46 chromosomes. You saw how you could divide 46 and get two 46s. But now from 46 you will get 23s, right? And it turns out you will not do it in one stage, you will do it in two stages. So from 46 you will get, as the end result, you will get 4 23s.
Oh yeah, 2 cells then.
Because if you just did mitosis, you would have still got
Reverse mitosis.
two 46s, right? But instead of two 46s, you'll get 4 23s.
Wait.
Four
But
haploids.
if there's 46, how is there 4 23s? I don't understand that.
First
Each
Okay.
multiplies.
If you just did mitosis, it would have been 2 times 46, right?
Yeah.
Right?
Oh, because they multiply. Oh.
Yeah.
And then-
Everything, the reduction also happens through a multiplication.
Yeah.
Yeah.
Okay.
Okay? So remember these terms, haploid and diploid. And the process of going from a diploid to haploid is called meiosis. And the process from going from a diploid to another diploid is mitosis. Mitosis is cell division, meiosis is reduction and forming this special kind of gamete cell, haploid cell, which has only half the chromosomes. Yes.
Are haploids the same thing as gametes?
Yes. Haploids refers to
It's a type of
the number of chromosomes that a cell has. So diploid is like 2 times 23 because it's paired. Whereas gamete is physically that cell, the special cell which is formed as a result of meiosis. Okay, so there's a difference, but they kind of occur together. Okay. Okay, so meiosis. The good thing about meiosis is there's only kind of one mat in this. It's the P mat. So it'll still go through the exact same terminology. And meiosis is broken up into 2 phases, meiosis 1 and meiosis 2. So what happens in meiosis 1 is, so it still goes through the growth phase, and in the S phase, now what happens is, it still forms the sister chromatids, so, and then the coiling still happens, yes, Sriya? So, when it enters prophase 1, so meiosis 1, let's do meiosis 1. So this is prophase 1. Let's see who remembers what happens in prophase 1. Anushri.
So the chromosomes coil up,
Okay.
and then the centrosome separates, and then the nuclear membrane disintegrates.
Perfect. And exactly the same thing happens.
Okay.
Okay? So it's, in that sense, just like mitosis. So you can see these from the coiled state, you see this, these are the chromatids, this is the centromere, and so these are the sister chromatids. These are the chromosome pair. I've drawn them together. And then, let's say it's two organisms, and it's like this. So this is one chromosome pair, and then this is another chromosome pair, okay? Prophase 1. Same thing. Metaphase 1, what would have happened? Yes?
The chromatids or chromosomes would line up in the middle, and then the
Good.
spindle fibers.
Good. And so the centrosomes have appeared, and then they line up. But now something different happens. Okay? So and I'm going to draw this. They still line up in the equator, but now the two copies of the chromosomes which have the same genes in them, they line up side by side. Okay? And the threads, earlier what was happening, the thread would go here and a thread would go here, and they would pull it together, and you just get two copies, right? But now the difference is the threads would go here, it would go here, and it'll still push. Very similar to mitosis, but instead of pulling the chromatids apart, they are pulling the chromosomes apart, and this is really where the reduction is happening. Okay? So, metaphase 1, this happens. Anaphase 1, same thing happens. And so now, instead of having 46 different chromosomes, how many chromosomes would you have on the two sides? Anushri? 23. That's right. 23, but what is the difference between this chromosome and what happened in mitosis? Apart from the fact that one is 46 and the other is 23? Yes, Srijan?
They are identical anymore.
No. No. There is a sister chromatid. Remember, the sister chromatid didn't exist in mitosis because they were pulled apart, right? Okay.
I know.
And now something else also happens which is going to explain Mendel's laws, and this is very fundamental. This is very, very fundamental. So please pay attention. Yes? What's the question?
I think I remember what actually happens. The difference. It was that in mitosis, there were just two chromatids.
Yes.
But now, instead of that, there will actually be 4 chromosomes.
No.
Because
Okay, the same picture at this point in mitosis would have looked like this. So if this was, let's say these lined up like this, okay, which is not true. In mitosis, they don't line up like this. They would have lined up kind of in the equator completely. The difference is, in mitosis, one sister chromatid would get pulled in this direction, the other sister chromatid gets pulled in this direction. So at the end, you have a chromosome which doesn't have a sister chromatid. It doesn't look like this. It looks just like this. Just like this. Right? You get that, Srijan? That's the difference. And now there is some other magic that will happen. And this will actually explain to you Mendel's laws. Two things. One is segregation. Remember segregation? Who remembers segregation? Yes, Naomi.
Segregation. I have it in my notes somewhere. Okay, it might be in here. Okay, here it is. Here. It says that the alleles that make the genes separate from each other or segregate.
Good.
So,
Good. That's exactly what happens. So imagine what we said. The genes are actually a part of the chromosome, right? So let's assume that the gene for tallness, okay, was present here. Okay, actually, I shouldn't have used red. Not that I have much choice here. Okay. So I'm going to try to do this properly. So let's say the gene for tallness was right here, okay? And so these are sister chromatids. So the gene for tallness would be right here. So actually let me blow this up, okay? Because this is, even I can't see properly, so I'm sure you can't. Okay, so let's focus on one chromosome pair, okay? Okay? So and pretend that this is the gene for tallness. Now, these are sister chromatids, so they have exactly the gene in the same place, same exact allele, right? But now, this one, it's a pair, right? It has the same genes, but this one says, let me get another color. See, this is tall. This is the recessive version, okay? In that Punnett square, this is what we wrote, right? This is physically where it's present. This stretch of DNA, which signifies the gene for tallness, you have two different copies, okay? And now, what is happening? They are being pulled away from each other, right? So, one side which has 123 will have this one, but the other side will have the other one, right? So now the two cells are suddenly different. They have the same genes, they have same number of chromosomes, but the alleles may just be different. And this is segregation. This is Mendel's process of segregation. You get it? And then there is another magic that happens when these two things line up together. It is called recombination. Okay? And in recombination, what happens is the genetic material here sometimes goes over to the other side. So they just exchange. This exchange doesn't happen at random places. They happen exactly at the boundaries of the genes. the material from one side goes to the other side. Yes.
What is it called again?
Recombination.
And how does it work? Like, how does one gene from this side go to the other side? That doesn't, how does it happen?
Basically these things, when they come close together, because of the proteins that are there, they kind of, remember, they were all like hair-like things, right? They just break away from each other, and then they just go and attach to the other side. I mean, do you want to add anything more to this? Because
Yeah, what you said is right. There is, it's called homologous recombination. Basically, the sequences are identical in the DNA, right? And there are proteins that can catalyze exchange of the different DNA strands. Okay, this one strand can invade the other strand, and they can exchange the genetic material. So you restore the chromosomes, they're exactly perfect, but there is some exchange of genetic information. Okay, that happens whenever they line up in the center, next to each other.
But the capital T always comes together, always come as a pair.
always comes as well.
Yeah, it won't happen like when they exchange and capital T and small letter T come as a pair.
No. So exchange means this goes here,
The
this
entire
goes here.
gene won't change, it's very rarely you change, really small segments are changed sometimes. That's very rare, I think usually the entire phenotype is maintained.
Now, other things also may happen. There could be mutations that happen which create a completely new allele. A new gene could be created if something abnormal happens in this process, and that's one of the ways we evolve. Most of these changes, if they don't follow these two things, this recombination, the normal recombination, they result in death. I mean, these are usually mutations are usually lethal, but once in a while there's a good one, and sometimes new genes can be formed through this process of recombination if it's not perfect. But so what this tells you is that sometimes you can get one haploid, one gamete can get one version and the other gamete can get another version. And there is no guarantee that this would be an identical form of what was originally there because of the recombination. Now, just to give you an idea of what this means, is, say in human beings, okay, there are 46 pairs, right? And we are getting 23 on both sides. And each of them can happen in how many ways? 2 ways, right? I mean, either, like, if I look at these two at the end, 23 and 23, let's say A1 and A2 are the 2 copies. So this could get A in 2 ways, and correspondingly this could get A in 2 ways, right? So the total number of variations that can happen as a result of this segregation is
46.
what? How many?
46?
No.
Wait, so for,
For every chromosome, there are 2 ways by which it can go, right? Either one can go in this direction, the other can go. So it's how many different combinations can you get? Okay, just think about it. It's going to be 2 to the power 23 because you can get either one copy here or the other copy here, right? So, right?
Yeah.
This is just think about it. So just by the process of segregation, there are 2 to the power 23, which is a rather large number. It's actually 8 million. Yeah, it's about 8 million. Different combinations can be formed. Now on top of that, usually there are about 30-40 recombinations that happen in this process. Now you can see how many can happen, right? Because each of those 30, 40 could lead to a different version, which is different from what existed before. And so it goes into many billions, okay? So this is the reason why once you go through this process of meiosis, you basically end up with something which is completely different. That this is, and we'll come to that later, but there are many different gametes that can form as a result of going from 46 to 23, this reduction, and the fact that this recombination happens. Is that clear? Billions of combinations that can happen. Okay? Yes.
If there are billions of combinations that can occur, then when suppose Gregor Mendel was testing it now, like if this was true now, then
It's
what-
still true now.
then how would that explain that? Because there will be only.
He was only looking at one characteristic, right? He was looking at the tallness. And it so happened that the tallness happened in one segment, and he never saw any recombination. He actually always just, so there's a 50% chance that you either get this one or you get the other one, which is exactly what we do in the Punnett square, right? This is how you relate the Punnett square back to what is physically happening during meiosis. Because if you happen to get one of the gametes which contain this particular chromosome, you get the tall thing. But it's equally likely, because two gametes formed as a result of this, it's equally likely that you could have gotten this other one, right? And then you would got the recessive form, right? Is that clear to everyone?
Each gamete is very unique because of the combination
Yes.
of all the different genes. But when you look at one character, there are only potentially 4 types of gamete. You have 2 with big T's and 2 gametes with small t's. So when you look at one character, it's always Mendel's laws. When you look at the entire combination, each gamete is unique. That's why each one of you are so different. You don't look like your brother or your sister, right? Because of all these varieties of combinations that can take place. So Mendel's law is fine. There's no problem with that.
You understand? This is fundamental. It should be very clear to everyone. And then, after this, it goes through the anaphase and telophase, very similar to mitosis, except for the fact that what is being pulled apart is not the sister chromatoids, but actually the entire chromosomes, which still have the centromeres attached to them. And then, so now you have 2 cells or 2 nucleus at the end of telophase 1, which have chromosomes with 2 sister chromatoids, but now half the number of chromosomes. And now guess what you need to do to get it back to something which doesn't have the sister chromatoid. You follow what happens in mitosis, except that you are working on half the chromosomes. And that is called prophase 2, metaphase 2, anaphase 2, telophase 2. And if you look at the pictures, it will now all make sense. And in the second process, on half the number of chromosomes, where you have the sister chromatids which are exactly identical to each other, they get pulled apart. And now you, out of one cell, you have formed 4 cells with each having half the number of chromosomes. Now tell me something. Are these, how many out of these are identical to each other, out of these 4 cells?
If there is, well,
two each are identical, right? Because the second phase was just like mitosis. You just pulled the sister chromatoids out and you got these final end result. So these two are identical, these two are identical. But this two and this two are different. Because of what two things? Segregation and recombination. Remember, segregation is the fact that you don't know out of the two chromosomes, the chromosome pair which carry exactly the same gene, which one goes where, right? And then recombination is the fact that there is exchange of genetic material that happens here. So apart from the segregation, these two could just look different from what they originally were.
So then nothing will be identical.
I think,
Right, I mean,
If
recombination takes
recombination
place-
happens in all the chromosomes, then nothing would be identical.
Even if it happens in one, it will be different from the other. So it
Exactly,
doesn't
but the
matter.
other chromosomes could be the same.
Yeah, so all four are unique in some
Yeah.
ways.
Exactly. Yes.
yeah, because I think the Hamas the hybridization happened in the close nearby
Yes.
chromatid, not the outside ones.
No, not the,
Yeah, only the central ones that are.
Only the central
crossover,
ones, yes.
And then they exchange material. The outer
Oh.
ones remain intact.
oh, the outer
So,
ones remain
Yeah,
intact.
so what happened is you got 4 unique ones.
Oh, actually, then you'll get 4 unique ones,
Yeah.
yes.
Yeah.
So, sorry, I stand corrected. So, did you get what?
You should draw that maybe, then
I'll
people will
watch the
understand.
video.
Sorry, yeah, you'll watch. Is the video useful, by the way?
Yeah,
Okay,
I can see basically exactly what it is in class, so.
okay, good. Okay, so I think that's a very good point.
Draw the crossing over, then it
So the
makes-
crossing over does not happen, so after the crossing over, this is how it will look like. After the crossing over, so now this would become green, this would become yellow, this would remain green, and so if you see these two chromatids remained exactly like this, but the crossing over happened here. Okay, so you actually end up with 4 distinct copies, not two which are identical. The crossing over did not happen here. That's a good point, actually. Yeah. Shows I'm not a biologist. Okay, and then the meiosis 2, you get the 4 cells. And so now, if you think about it, from one parent, you are getting basically one out of the pair of the chromosome, right? And even this diploid organism must have gotten this from its parent, right? What does that mean? It basically means that out of the 46 chromosomes, so now what will happen is you'll have one gamete coming from one of your parents which has 23 chromosomes. You'll get another gamete coming from another of your parents which has 23 chromosomes. And that's how you get the 46 chromosomes, right? So that means half of your genetic material comes from one parent and the other half comes from the other parent. And right now we are looking at what is happening in one parent, right? And this parent must have got one from his one parent and his other parent, right? So that means you get 1/4 from your father's side grandparent, 1/4 from your mother's father's side grandmom, and 1/4 from your mom's side grandfather and mom's side grandmother. That's the way it all works. And now this explains a lot of what Mendel actually saw. So if you just imagine that the tallness was present here, and maybe the roundness was present in another chromosome, they would segregate independently, right? That's the law of independence that you study.
I think Mendel was lucky because the phenotypes he's looking at were actually on different
On different
chromosomes.
chromosomes.
That's right, they were all linked, then this theory will evolution.
And now if you look at the exceptions to Mendel's laws, that will also now make sense. There is something called linked traits. Because if two genes happen to be very close to each other, they will always go together, right? Or especially they are on the same chromosome. Even recombination, unless there is recombination, they'll always go together, right? So you can actually now look at both Mendel's laws and the exceptions and see why this happens the way it happens. Okay? Any questions? This is very fundamental, okay? You need to understand this. And in Science Olympiad, the questions would be, they will show you some picture and ask you, is this meiosis happening or mitosis happening? What phase is it in? Yeah.
I have a question about like the Science Olympiad, like the actual thing.
Mhm.
So are we gonna all so each of our groups will look at the same questions?
Yeah.
Oh, okay.
I believe exactly same questions, and I think what you can do is you can even divide the questions among your teammate, and then you answer half, your teammate answers the other half, and then you check yourselves together.
So there aren't any like specific questions that like each group will have?
No.
Oh.
Each team will see exactly the same questions.
Okay, so basically, our like event will just be answering questions.
Yes,
Okay.
yes.
That's what I do since I don't have a partner.
You have a part.
No, Lydia didn't come.
No.
Oh my
She'll come on the event day.
God.
No, I think we need to sync up with her
And,
and you need to sync up with her offline, because I think we should start a separate email thread on that. But don't worry, nobody has started working together. We are still going through the material that we need to cover. Now another thing that you will see is called karyotypes. Okay? And so karyotypes is a picture of all the chromosomes that are there. And sometimes in the process of meiosis, some errors, both mitosis and meiosis, errors can happen. One of the errors is one chromosome just doesn't happen, it doesn't come in. Yeah. And sometimes you can have an extra chromosome, and we'll go over those are actually there. But now that you have this background, if somebody shows you a karyotype or an ideogram, you should be able to say what is wrong. So now, having gone through this whole meiosis and mitosis, I will just cover one more thing. This will not take a long time. This is determination of gender or sex determination. Okay? How does that happen? Yes.
Our 23rd chromosome determines whether we're going to be a male or female. So in either two X's,
which means you're female, and it's an X and a Y
That's
chromosome if you're a male.
That's exactly right.
Yeah.
That's exactly right. So in human beings, we already know that there are 46 chromosomes, right? And there are 23 pairs. 22 pairs are called homologous. Homologous means they have identical genes. There could be different alleles. Just because there are same genes, it doesn't mean that they are identical. There could be different alleles. But otherwise, they are the same. Okay? That's why they are called homologous pairs. So if you are a female, you have let's say this is they call it A1, A2. These are like the homologous genes. Homologous chromosomes. And then the 23rd one is not a homologous chromosome. It's called the sex chromosome. Okay? And that one, for a female, is actually homologous. It's X and X. It's got two X's. For a male, this is chromosome number 23. And this numbering is arbitrary. We just for convenience, we say this is the 23rd chromosome, okay? For a male, all this is just like a female, but the sex chromosome, it's got a X and a Y. And the Y is much, much, much shorter than the X. Okay, that's one way by which you can say whether it's a male or a female. Okay? So when you see a karyotype and they ask you, is this a male or a female? And they will show you the 23rd chromosome. Look at the sizes of the chromosomes and see if they look pretty similar, that's a male. If one of them is
Female.
it's a female. And if one of them is much smaller, that's a male. Okay? Now that you know meiosis and now that you know about this 23rd chromosome, tell me, when you have a child, is it the father's chromosome or the mother's chromosome that determines whether the child would be a male or a female, and why? I want somebody else to answer. Brandon, why don't you guess? Both? Okay,
The fathers,
the fathers. And why?
because his 23rd chromosome can change. Like it can be an XX or an XY, but the females is always an XX.
Okay, so, yeah, you're right. So, in the process of meiosis, what is going to happen is these 46 will get into what? Two 23s, right? And if you look at the 23rd chromosome, the two gamete or the four gametes that will be produced from one of these cells, two of them will contain what? X. And the other two will contain what?
Why?
No, it's a female.
Oh, female.
It's still X, right? Okay. So from the female you have a X and the other one is a X. Now, if you're a male, what would you get?
XY.
You could get a X or you could get a Y, right? And now the new individual, this is all chance. either this could fuse with this,
How does
And
the
then you
fusing
have what?
happen?
sorry,
How does the fusing happen?
how does the fusing happen?
Like how does one of the things fuse with the
You
other?
learn that, you learn that.
Sounds good.
It's not going to be covered in Science Olympiad. Okay. So, just assume that fusing happens. So when these two fuse, what would be the gender of the child?
Female.
Female. But suppose this fuses with this, what would happen?
Male.
Male, right? So that's how you have males or females in human beings. Because of this difference in chromosomes, the chromosome structure. And then, because of this, there's something else interesting that happens. Is it a question or
No,
a
it's a
comment?
question. It's a question.
It's a question. Okay, let me just finish this part, because this is, I just want to finish the flow. I'm in the flow. Okay. Now, you can imagine that the fact that males and females have a 23rd chromosome which is different. Suppose you had a gene in Y, okay, that was dominant. Would that, and that caused a disease, let's say, would that ever show up in a female? No, right?
That's a separate gene.
Exactly. And then, suppose there was a dominant trait in one of the X's, and it never went to the Y part of the male. Would it ever show up in the male?
Yeah. Oh, no.
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 No. So,
No, it didn't
that's
go
why you can have these they call sex-linked diseases. So the disease only happens in either the male or the female. And that is the reason.
Is there also like a disease where there's extra X chromosomes?
Yes, there is an extra, that's called a trisomy, and if one is missing, it's called a monosomy.
So what happens then?
You have all kinds of diseases. And I haven't read up that part, but if
For
somebody knows
example,
that.
Down syndrome is caused by an extra 21st chromosome. So it can have an extra chromosome that causes usually mental problems, or it can be lethal, the child will be dead. It's not all the way.
So if you have 3 copies, it's called a trisomy. If you have 1 missing, it's called monosomy. Anytime you see something different, it's abnormal, it's going to cause some problems. Usually nothing beneficial. Okay? Yes, now I'm open to questions. Devangi, are you done with your question? Okay, Srijan.
If you were to draw it in a Punnett square, how are you supposed to do it?
The Punnett square now, so the relationship is very simple, right? You have a pair of chromosomes, right? I'm just drawing it big. And this is the allele, this is the gene which is responsible for tall. And this is the, should be another color. This is another allele that's responsible for the recessive form of it. And you don't know which one came, right? So in Punnett square, what you did is, this is one of the parents, so you wrote it as T, and then you said, I could either be a T, and if this gamete happened to fuse, then this T would happen, and if this gamete happened to fuse, then this T would happen. So this is how the Punnett square, and then same thing from the other parent. You would have a T and a T, and you don't know which one you get. So this is how, this is the chromosomal basis of heredity. Okay? Is it all clear now? I haven't gone through any of the slides, but the slides are there in Google Drive, and I hope you can go through them yourselves, and then if there are any questions, then you can definitely ask in the next class. I think I have covered most of the material from which 2 sections of Science Olympiad questions come. So what I'll do is next week, this room is not available. So what I'm going to do is I'm going to kind of record the lecture and put it, and I'll cover the things that was not covered last time, like the more exceptions to Mendel's laws that I didn't cover very well, and then some of the disorders that happen as a result of errors that happen during this process of mitosis and meiosis. Because this is either a perfect copying machine or a random reduction machine. But you know, it's a physical thing. Sometimes with a very small chance, errors can happen. And when errors happen, you have maybe a child with Down syndrome, or you have a child with other genetic disorders. That's why it's called genetic disorder, because at the construction time, there's something wrong with the set of chromosomes that you have, and that just doesn't make things normal. Yes, sure.
What's Down syndrome?
What's Down syndrome? Actually, I don't know.
Down
It's-
syndrome,
Yeah, Down
it's due to
syndrome.
an extra chromosome, 21st chromosome. What happens is these children, their IQ is very low. They have IQs of 20 to 25, and they also have other features. You can see the facial features, elongated face, big ears. They have social problems. They cannot interact with other people, and usually they die by the age of 25, 30 years old.
That's sad.
Okay.